Hello. This time I'm going to deal with the another important enzymes that are used in the biotechnology or recombinant DNA technology, and the name of the enzyme is DNA ligase. So, what happens in the cloning process? In the cloning process, we are integrating the any foreign DNA of interest into the cloning vectors. Suppose that this is a cloning vector, double stranded, and this is the our DNA fragment. It may be either the end with the blunt or with the sticky end. And suppose that this is obtained by the restriction enzymes 1, for examples, eco R1. With the same enzymes, we will just cut this DNA also. And when this DNA will be cut, then it will create a gap. It will create a gap within which this DNA will be integrated. But after integration, there occurs a gap that is called the nick. So basically, the nick and gap, these two are different. Nick means there is no any loss of nucleotide. No loss of nucleotide will be there, and the nick is due to the absence of the phosphoester bond. But in case of the gap, both nucleotide as well as phosphodiester bond is absent. So this sealing is done by the enzymes that is called as the ligase. Perhaps in all the natural conditions, this ligase is responsible for doing so many type of the biological jobs, and that I will deal through the PowerPoint presentation. So here, this ligase this ligase is a protein enzyme, and definitely it is a monomer, and initially it is responsible for making the genome integrity. The ligase was first purified and characterized by the Gellert, Lehman, Richardson's, Hertzwirth in their laboratories. So in the recombinant DNA technology, any gene is integrated and it is ligated to make a continuous frame by means of the ligase. So we are using in this very cloning process three different enzymes. One enzyme is restriction endonuclease, which will cut the DNA, and another one is the ligase, which will seal the integrated DNA at their end, and third one is the polymerase, which will make the copy of this very DNA. So such DNA is called as the recombinant DNA or the chimeric DNA. So chimeric DNA is possible due to the involvement of these two enzymes, that is restriction endonuclease and the ligase. So this DNA ligase play a very important role in maintaining the genomic integrity by joining the bricks in the phosphodiester backbone of the DNA during replications, during the combinations also. Then it also helps in the repair of the damaged DNA, and in biotechnology, it is used to intake the DNA fragment into the vector. So in a layman, we can call them as molecular glue or the molecular sutures, similar to the molecular Caesar, which is restriction endonuclease, and the molecular glue for the ligase. So what could be the source of this ligase? Definitely, it is already mentioned that this ligase is of universal occurrence. That is present in all the animals, plants, bacteria, viruses like that. But for the purpose of the biotechnology or in the recombinant technology, we are using the two different ligase enzymes from the source. One is the E. coli and another one is the T4 bacteriophage. Besides, other ligases are also created in the laboratory to provide the more efficiency that I will deal in the next slide. So E. coli DNA ligase E. coli DNA ligase is the 75 kilodalton monomeric proteins, while the T4 bacteriophage is 68 kilodalton. And it was the first ATP dependent because, see, it is a thermodynamically active. Thermodynamically, it is active. So it will require the ATP, and when the temperature is lower, then the activity of the ligations is proceeded. So another enzymes which are used for the ligation of the DNA molecules or the RNA molecules. Here are some of the enzymes like DNA ligase from E. coli, from the bacteriophage T4 ligase, and another one is the ampligase, amp ligase, and this ligase is thermal stable. It is thermal stable because the natural ligase, it cannot be used for the ligations as in vitro condition because once it is uh, isolated, they get the denatured. So we have to choose that very ligase which can tolerate the temperature. So this is the temperature tolerant ligase enzyme. But sometimes what happens, the natural ligase, they just perform with the less efficiency in some of the cases. So in order to increase the efficiency, the ligase enzymes are engineered. It means that if the this very protein is there, so we have to just fuse some another proteins in order to achieve the efficiency. In order to achieve the efficiency. So this genetically engineered ligase enzymes is very much used. And next is the RNA ligase. This ligase used to just to join the RNA nick, the nick which is formed in the RNA. That is, for examples, in case of the anticodon loop. We are very much aware about that. When a translation process, when a translation process is proceeded, when a translation process goes on the surface of the ribosomes, then what happens? One tRNA, tRNA that charged with the amino acids, and it get translocated on the A side or the P sides of the ribosomes. So it has got the anticodon loop. Basically, it is clove leaf-like structures where it has got the anticodon loop. So suppose that it has got the ACU. This is the anticodon loop with the ACO anticodons. So what happens when this, sometimes if it breaks, like if it will be like this. So this cannot performs the pairing between the codon and the anticodon. So in order to avoid this, ligase will be there, and this will be the RNA ligase. So this RNA ligase comes and it just seal the DNA. So here this enzymes comes. So altogether, there are the E. coli DNA ligase, T4 ligase, ampli ligase, genetically engineered ligase, RNA ligase. Okay. But beside this, there are others ligase also, which are found in the different animals or the plant. If ligase is definitely an enzyme, so it must have its substrate. So what are the substrate to which it interacts? So I told you that ligase seal the nucleic acid fragments, so either with the blunt end or with the sticky ends. So various substrates are there. For example, if the DNA nick is there, either in the double stranded or the single stranded, or the DNA fragments with various length with the cohesive ends. Okay, this is the cohesive ends. One is this, another one is this is the cohesive ends. This is also cohesive ends. And DNA fragments of the blunt end. If the blunt end will be there like this, if the blunt end will be like this is the blunt end. So ligase can seal all this type of the DNA fragments. So beside this, it can also make a hybrid. Suppose that this is DNA, this is RNA. This can be also ligated. This can be also ligated. Okay, so it can also seal the DNA RNA habits. For a purpose of comparison, so DNA ligase or the T4 ligase, here are some of the comparative points. For example, the DNA ligase, it is present in all except viruses, but only E. coli DNA ligase is used in the biotechnology. While the T4 ligase is obtained from the bacteriophage and it is the most commonly used more than this DNA ligase. Another major difference is that it uses a cofactor. So here the cofactor is NAD, nicotinamide adenine dinucleotide, and here the cofactor is the adenosine triphosphate. When the ligation comes, so it cannot ligate the blunt end DNA. It is only ligating the cohesive end DNA. But here it can ligate both cohesive as well as blunt end of the DNA fragment, as well as the RNA DNA hybrids, but not the single stranded nucleic acid. Single stranded nucleic acid, if it has to be ligated, it cannot ligate it. If it is the RNA or the another RNA, it cannot be also ligated. So for that purpose, we have to choose the RNA ligase. So the only the limited substrate is there. That's why it has got the narrowness activity. And here many substrates are under the action of the ligase, so the broad activity is there. Also join the single stranded nick in the double stranded DNA. So if it is the double stranded DNA with a nick, so it can also be performed by the T4 ligase. If it is the DNA, if it is the RNA and there is a nick, then that can be also performed. So we can make a chart like this is a double stranded DNA, double stranded DNA with nick. This is RNA DNA hybrid, RNA DNA hybrid with a nick, hybrid RNA DNA with nick. Then if the if it is blunt or if it is DNA and RNA or it is RNA or RNA, so ligase will be there. Ligase. So ligase can perform all these type of the sealing, but comparatively, this is this cannot be performed by the single standard cannot be performed by the T4 ligase. This cannot be performed by the what is called E. coli DNA ligase. So it will be only E. coli DNA ligase cannot ligate this blunt end. It will be ligated by the T4 ligase. Next, I told you that when the efficiency is less, so for that purpose, we have to genetically engineer the DNA ligase, and for that purpose, we have to add some proteins part like either the P50 or necrosis factors, KB, and which just get fused. And when this was fused, the efficiency was found to be improved as 160% or more active in case of the blunt end ligations as compared to the wild type. So, how this efficiency can be improved? Because you see, the two types of the ends are produced by restriction endonuclease. Here, eco R1, which produces the sticky ends, SMA1 produces the blunt end. So, blunt end, if it will ligated, then it will be the having the less efficient, and it will need more concentration of the ligase, and sticky end is efficient. So, problem is always here how to ligate the blunt end DNA, because here we have to use more ligase. So, for that very purpose, we have to use the enhancers. For that very purpose, we have to use the enhancer. So when a ligase just bind with the substrate, the ligation process is proceeded under the three different steps. First is the adenylations, adenylation of the enzyme, that is addition of AMP in the active center of the enzymes. Second, release of the pyrophosphate and transfer of AM to 5' P, so called donor, and formation of the pyrophosphate bonds. And third is the formation of the phosphodiester bond. So the three adenylation process, then release transfer of AMP to the 5' phosphorus group, and third, the formation of the phosphodiester bonds. So these three steps is for the DNA ligation and T4 ligase ends to end joining. This could occur inter or intramolecular, but intermolecular joining is correct. And ligation requires ATP, and in the laboratory condition, it is always carried out at the 4 degree centigrade temperature because it lowers the kinetic energy. To reduce the separation of the paired sticky end, which is later stabilized by the ligation. However, long reaction times are needed to compensate the low activity of ligation in the cold. So here it is the representations of the mechanisms. You can see here the NAD, it is a cofactor in the E. coli ligase, ATP for the others. So in the first step, adenylation will occur, sir. That is, the enzyme will interact with the AMP. So here you can see this ATP, it is broken out into the AMP plus 2pi. This inorganic phosphate is released. Okay. And after that, this enzyme linked AMP, it gets interacted with the 3 prime OH. This is the 3 prime and this is the 5 prime. Okay. So adenosine will just interact with the this one to form the pyrophosphate bonds. Pyrophosphate bonds. And then the nick is sealed by the nucleophilic attack, and the ligation is occurred here. So this is the complete ligation sum. So one is the adenylations, this is the adenylation process. Then here is the transfer of the pyrophosphate, and third is the ligation sum. So these are steps under the mechanisms or the catalytic action of the ligase. This is the unit to measure the activity, and it was named after the scientist Bayes. So, in the earlier slides, I just explained that to enhance the ligations, to increase the ligations, or to have the more confident on the ligations, we have to use the enhancers. So, one of the enhancer that I told you, it is the genetically engineered ligase. But beside that, the DNA polymerase 1 and the polyethylene glycol. This is 1, this is 2, and this is the 3. These 3 are used as the enhancers. For example, the activity of the E. coli DNA ligase can be enhanced by DNA polymerase at right concentration. Enhancement only works when the concentration of the DNA polymerase 1 are much lower than the DNA fragment. See, condition is there. The DNA polymerase 1 should be in the low concentration, otherwise it must may have the adverse effect. Similarly, for the T4 ligation, T4 ligations, the enzyme concentration is kept high, and the polyethylene glycol is added to reaction mixture for stimulation. So then polyethylene glycol that stimulate the activity of the T4 enzymes. In this way, the genetically engineered ligase, DNA polymerase 1, and the polyethylene glycol, these act as the enhancers. So, blunt end ligations. Of course, the problem always comes when the two, the two DNA fragments, having the blunt end, if they ligate, the ligation will be temporary. Ligation is temporary. So how to make it permanent? So for that purpose, we are using the two different molecules. One is called as the linker, and another one is called as the adapter. Linker is the double stranded DNA. Linker is linker is double stranded DNA. Linker is double stranded DNA with one site, that is the restriction site. So this is the restriction sites. So this is the linkers. Definitely it is 1 to 14 base pairs. This is the synthetic one. So linker is the synthetic one which possesses the one restriction sites and the sequence that varies from 8 to 15 base. So in this case, the blunt end DNA is added. Okay. This is the DNA fragment with blunt end. So how to make this blunt end sticky? So once this is obtained, the DNA fragment that gets associated with the linker. So after the cleavage by the restriction endonuclease, this is the site, because this site is a eco R1 site. So when it is treated with this eco R1, then what happen? Then there will be the formation of the, this is of course the blunt end DNA, but here this is like this, this will be, and this will be like this. So it will be having the sequence C C G A, C C G A, because here is the cut, and here the sequence is A A T T, then C G G. So now this blunt end, it has been generated as the cohesive end. Now the ligation can proceed. So in the on the both sides the linkers are added. If the both side linker will be added, then definitely it will generate the sticky end. And the adapter is the it is the cohesive ends. Adapter is the cohesive ends. So this is the adapter with cohesive ends. When the blunt end DNA will be there, then this adapter can be added. So now this becomes the sticky. So adapter is with cohesive end, linker is with the blunt end with possess the restriction enzyme site. So here is no restriction sites are present in the adapter. So you can have the linker and the adapter differences. So here I am showing that how the linkers and adapters can work together. Another problem is that if the ligation occurs, then there is a recirculation of the plasmid DNA, because once you cut this plasmid by the RE, then it will create like this, but soon it has got the ability to recircularize, resealing. So how to prevent it? So you know that one is the P, this is the OH, then this is the P, and this is the OH. Okay. So if you remove this phosphorus from this very sites by using the alkaline phosphatase, then what will happen? These two will be having the same end, OH, 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 OH. Now it cannot form the phosphodiester bond. As you see here, this is the P, OH, P, OH, alkaline phosphatase, then both OH, and it will prevent the ligation. There will be no ligation at all, no reactions. So what will happen? You have to add again the POH, phosphoric acid, and this will just add the ligation. Ligation process will occurs, and there will be a nick, and that nick can be sealed with the help of the ligase enzymes. So all these problems occurs during the ligation process, that is the deficiency of the efficient ligations, then recirculation of the ligations, then the blunt end ligations. So all this can be treated as the problems, and there you can just increase the concentration of the enzymes, or you can use the enhancers. So at last we have got the key. What are the some of the importance or application. So DNA ligase is an important enzymatic tool without which RNA cannot be achieved. The important functions are involved in the ligation or joining of two fragments, like two Okazaki fragments in replication. The fragments are formed on the lagging strand, but after its removal, but after the removal of the primer, the DNA segments join together to give a continuous strand of the. Okay, always this is performed by the 3' OH and the 5' phosphorus. It is used in the ligation of DNA in the vector for cloning and expressions, used in ligation of linker and adapter at the blunt site of DNA fragments. It is also used in ELISA to form the enzyme substrate conjugate. For example, the p-nitrophenyl phosphate is a substrate for alkaline phosphatase but cannot conjugate. So if you remove this phosphate, if you remove this phosphate, then the ligation will be there. So AP remove the phosphate and then it forms a conjugate. Self ligations is prevented by use of the alkaline phosphatase, as you see here. In the dephosphorated vector, DNA cannot be recirculated in the cloning procedure and can remain linear for ligating with the other DNA. Ligase requires, of course, the 3' OH and the 5' PO4. So thanks for the watchings, and at last I must say thanks to you. Thank you very much.